everyone, this is 101 Class Survival Guide, and I'm back with another video on all methods for nucleic acid sequencing, and this is the part 2 in which we will be discussing about automated Sanger sequencing method. The automation and sample preparation for the Sanger sequencing method was performed by Fred Sanger in 1977 that utilizes the chain termination method of the previous Sanger sequencing method and an automated form of detection method. The components for automated Sanger sequencing are all the same, including the ssDNA template, DNA primers, clinofragment, and the normal dNTPs. However, the ddNTPs for this type of Sanger sequencing, automated Sanger sequencing method, are dye-labeled. The dye-labeled ddNTPs are all of different colors. For example, ATGC, ddATP, GTP, CTP, and TTP are attached with 4 different colors of dyes. These dye-labeled ddNTPs, when passed through a laser beam, they emit color, which helps in their detection. The steps for Sanger's automated sequencing method are simple. First of all, we generate the dye-labeled ddNTP segments of the DNA, and then the segments are separated on the basis of size through the column. The smallest segment travels the farthest and quickest. The laser beam passes through each segment. The light emitted for every dye-labeled nucleotide is recorded and the sequence is noted by a detector. These are those 4 dye-labeled ddNTPs. Each one is labeled with a different fluorophore: yellow, green, red, and blue, so that when the laser beam passes through them, they emit color or fluorescence once. And each different type of dye-labeled ddNTP is incubated with normal dNTPs, and after the generation of different lengths of DNA segments, they are passed through a column and are separated on the basis of their size. Along that, a laser beam passes through them, and the emitted color is detected by a detector that sends the information for every emitted color in the form of the attachment of a different type of ddNTP or the presence of a different type of fluorophore, and each fluorophore's color represents a different type of ddNTP. This is how the results of automated Sanger sequencing appear on the attached monitor or computer. It not only notes down the sequence of the nucleic acids, but it also notes down their strengths, and it helps us in detection of the sequence along with their strengths. This was all about automated Sanger sequencing method. Thank you for watching. Please subscribe for more and comment below for queries or any future requests. The next video will be about para sequencing.